Hello everyone. I hope I'm audible. We'll wait for a few minutes for everyone to join and then start. All right, so let's start. Welcome to this live interaction session of week 7. Uh, so in the last session, we saw few bits of hyperspectral remote sensing, and in this session also we'll continue to learn about few more concepts in hyperspectral remote sensing, and we'll solve a couple of problems from previous year's assignments, and then we'll look at a small introduction to GIS. Right. So, from the last class, from the last session, we saw that hyperspectral remote sensors, they collect image data simultaneously in a large number of bands, like in dozens or in hundreds of narrow, small adjacent bands, and they generate a continuous spectrum for each cell, right? So here we see that this is this is an image, and these are n number of bands which are spanning spanning small narrow ranges in this spectrum. So each cell, so suppose if there are some 200 bands, so each cell will have 200 number of data points. So for each cell, we can generate this spectral reflectance curve like this, right? So a set of brightness values for a single raster cell position in the hyperspectral image, and then this is the plot of brightness values versus wavelength, showing a continuous spectrum for the image cell, which can be used to identify surface materials. Right. So when so we saw this in case of multispectral remote sensing also, that when the sensor is recording uh, the reflected values, in that case it is measuring the radiance, right? And uh, this radiance, it differs from the actual surface reflectance values because of contributions due to uh, source sensor geometry, atmospheric errors, terrain effects, and so on. So this radiance needs to be converted to reflectance first, in order for us to be able to uh, compare with the spectral reflectance spectra of materials on the surface. Yeah, so after converting it to reflectance, these values can be compared with field or laboratory reflectance spectra to recognize and map the surface materials. Right. Now, these measurements we are taking from satellites. On the ground, we use spectroradiometers. These are used to make ground based or lab based measurements of light reflected from the test material. So, this figure on the right, it shows the working of a spectroradiometer. So, uh, this is this is a single cell on the ground. So, light from this cell is being passed through a scan mirror and other optics. And then this assembly, it uses an optical dispersing element such as grating or like such as a prism. And then uh, the light which is reflected from these objects on the surface, it is made to pass through this prism, and then it splits into multiple wavelengths. So this prism, it splits the reflected light into many narrow adjacent wavelength bands. Right. And then afterwards, energy from each band is measured by a separate detector. So here, for each small band, we have a different set of detectors to measure the energy in that narrow wavelength range. So higher the number of detectors, higher will be uh, the total number of bands in that hyperspectral image. Right. So next is plotting a spectra in the spectral space. So yeah. So first we are measuring a hyperspectral data. So we are taking generating a hyperspectral image of from using remote sensing instruments. And there are spectral libraries which contain uh, spectra of which contain ideal spectra of various land cover types on the surface. So these spectral libraries help us in the interpretation of the hyperspectral images collected using remote sensing data. So now there are many such different spectra which are present in the spectral libraries. So suppose for a pixel, I have obtained a particular type of spectra and I need to identify the material which is causing this type of spectral characteristics. Now, if I were to compare this with each and every uh, in order to identify my spectral signatures, I need to compare it with different materials in the spectral library. So, how do we actually compare and discriminate between these large number of spectra? So, there can be multiple ways. So, either the spectra with minimum difference in the reflectance value from the spectrum libraries. So if we need to find a spectra in the spectrum library which shows minimum difference in the reflectance value from the image spectra, or there can be other method, there can be other different approaches like plotting a spectra in the spectral space. So in this case, uh, here each spectral band has a corresponding spectral value. So here we consider if in a hyperspectral image we have n number of bands. So right. So this is a hyperspectral image containing n number of bands. So this is one of the cells in this image. So there would be some reflectance value associated with this cell for each band, right? So here on the right hand side, we are given example with two bands in a two-dimensional space. So here on the x-axis, we have reflectance value in band 1, and on the y-axis, we have reflectance value in band 2. So suppose this is band 1 and band 2, right? So for this pixel, I'll have a reflectance value in band 1, and for the other pixel, I'll have a reflectance value in band 2. So these reflectance values are plotted against each other. So here we have reflectance in band 1 against band 2, and then we get a point, right? So this point is uh, is the representation of this spectra in a two-dimensional space. So if we increase the number of bands, then so 3 4 5, so in more in n dimensional space we can represent each spectra as a point. Now this makes it easier to compare it with the um, spectra from spectral library. So if my image has n number of bands, right, then I should pick an image from the spectral library which also has same number of bands. Then I will plot the spectra from spectral library as one point, and uh, my cell as another point. And then, depending on the closeness of the 2 points, we can tell if there are similarities uh, between the two spectra. So instead of individually comparing each and every spectra, this method allows us to compare different spectra in a spectral library with that of the actual image uh, in a more efficient way. Right. So we consider each band as one dimension and then plot uh, each spectra as a point in n dimensional space. Right. So uh, if two spectra, they have similar shape but different reflectance values, then they will plot along the same vector. So the vector joining origin and the point will be the vector representing that spectrum. So each spectra, if they have similar shape and different reflectance values, then plot uh, they will plot at the same point along the same vector at at different locations, like this. Um, I'll be back in a minute. I need to charge my laptop. Okay. Thank you for waiting. Uh, all right. So we saw that.
And a small clarification.
Huh.
See, when we say with the same, I mean like same what you're saying, the value reflectance value changes. Maybe it increases or decreases, but the shape of the graph would be same. That means when we say in terms of vector, right, the magnitude changes, but the direction will be same, right?
Right.
Okay, when we plot the vector on a graph for the
Hmm.
same values, then I mean, like the arrow mark or whatever the vector we form, right? Probably the reflectance values goes on increasing, but the arrow direction would be in a forward direction, at the same direction.
Yeah. So see here, in a case of this spectra, suppose this spectra represents this point A, right? So here, uh, so if this is A, B, C, so for C, I have higher reflectance values.
Right.
So it will plot along the same direction, but with higher reflectance values. Like it
Okay.
will plot
So if we move
away
on
from the
plotting
origin.
more dimensional spectra values, right? So arrow moves forward in the same direction, right?
Right. So this is in a two
Yeah,
dimensional
yeah,
space. Huh.
yeah. And one more thing is, see, when we are saying s- for with respect to spectral hyperspectral bands, right? So probably
Hmm.
like it will be in hundreds, right?
Right.
As per. So for when we take a pixel, right, on an image, exact
Hmm.
image, the target
Hmm.
image, so it will have underneath it will have 256 what do you say, bands and then the same pixels at the same location.
Right, right. Every pixel will have like hundreds of bands, and there are so many pixels in an image. So we'll see
So
what to do with the computation problems and all.
one clarification here. So for that, right? See, is that the reflectance value of each band will be differing, or it will be same for that pixel?
Uh, for each pixel, okay. So,
So let us suppose we have 300 band hyperspectral image,
hmm,
and then we have taken a pixel called 2 by 2 pixel,
hmm,
and two second rows second column pixel, and
hmm,
for that 2 by 2 pixel, right, we'll have 300 like I mean like band pixels,
right.
right? So let us suppose if band 1 pixel have reflectance value of 80,
Hmm.
band 2 pixel will have the same 80, or it may be differ.
No, no, it will differ depending on the wavelength and how the material reflects in that wavelength region.
Okay. Okay. So I
Right.
mean, like each band will be okay, for on based on the bands, it will be like will make those spectral bands, right?
Yeah, so depending on our band
Wavelength.
wavelength, the reflectance value will differ for every cell. Like
But it
suppose
will but it will be in the same range, right?
same, no,
So
it
let us
can
suppose
differ
if we
also.
take
Like
visible range. So visible range will be same, but only within that range the reflectance value will be
hello, uh, you're not audible.
sorry, my connection I lost.
Okay,
I'm
okay.
back again.
Huh. So you were telling.
See, let us suppose visible range. Let us suppose, just for example, we have between 4 mm to 6.5 mm or 7 mm, right?
Hmm.
So if you take image within that range, right,
Hmm.
bandwidth range, uh, let us suppose we make an image with 300 bands. So
Hmm.
that range will be divided into that wavelength will be divided into 300.
Hmm.
Is it right? What my understanding?
Yeah, so suppose my hyperspectral image is so my total wavelength range is now let's just look at this figure on the left,
Okay.
right? So
Yeah.
here my wavelength range is from zero point 0.4 to 2.4.
Yeah.
So suppose I have 200 small bands in this range. So if I were to divide this, then my each band will be like this small or even smaller than this.
So 200 equal wavelengths will
Right,
be formed.
right.
Okay. Okay.
So now my pixel. So this pixel can be anything. Like now on left hand side, I have uh, granite or basalt or concrete, right? Or before we saw plots like water, soil, vegetation, and so on. So this pixel can be anything. So this pixel, suppose this is a granite pixel,
Okay.
right? So depending on the composition of that pixel, my response in each band will be different. Like
Okay.
now if my pixel is granite and my first band is suppose this, right? This are you able to see it?
Yeah.
The small band. Huh. So here the response, the reflectance value will be somewhere around this, like maybe 30%.
Okay.
Then in the next band, it's increasing, like the reflectance value is increasing in the curve. So in the second band, it will be somewhere around here. Hmm. Yeah, in the second band it will be around here somewhere. In the first band it was here. Then in the third band it will be somewhere here. Next band like this. So for every cell in different bands, the reflectance value will be changing depending on how that material spectral response is.
Okay. Okay, so when we see an image, right? But
Hmm.
on the when we see an image on the screen,
Hmm.
we'll be seeing the top layer pixel, right? I mean, like the
So
radiance. So is it something like this the reflectance of each wavelength band will also will be able to see on the screen, or like it's only the band one, because band one would be the topmost pixel radiance, right? So that only will be visible on the screen of the target image.
You will be opening this image in GIS or
Right.
some software, right? So in GIS, you can actually change what type of like what bands you are looking at. So every point will have like total number of data will be like every point will have
No, I mean not on
as
the
many
graph.
values as the number of bands.
No, not on the graph. The naked image through naked eyes. I mean to say.
Uh, Through naked eyes, I'm not sure. It will depend on how we choose to represent the image.
Okay.
Like
No, just
it's
if
not
let us
necessary
suppose.
that we have to show first band only. We can show like a composite of three bands, or like we can uh, use
No, no. Hope
combinations of different
I think
bands and show.
hope you got my question. See, let us suppose we have a on our laptop we have our photograph. Okay,
Hmm,
we
hmm,
just right click and then open it, and it will
hmm,
opens up on the screen.
hmm.
So when we open it, when we are able to see our photograph on the screen, right? So the pixels what we see on the photograph
Hmm.
would be of all the band, sorry, wavelength reflectance, or it's only on the a specific wavelength band reflectance it would be.
Okay, so what pictures we see on the screen, right? So if we are looking at a true color composite, in that case, it's not like only one band. It will be so we have RGB color
Right,
components, right?
right,
So I can choose to represent any band in my red color. So
right.
I can represent band 4 in red color, band 3 in green, band 2 in blue, or I can choose which band to represent. So it will depend on what information I want to show. If I want to show some information that I want to show that granite looking dark in the image, right? In that
Yeah.
case, I will show my band one, like band one data. If I want to see like in vegetation, we have high reflectance in near infrared. If I want to highlight that vegetation in my image, then I will select those bands which have high reflectance value for vegetation.
Okay.
So it will depend on how I want to represent my data. I mean,
Okay.
this is as far as I know.
Yeah, yeah. Just I was just trying to understand the inner side things. That's the reason I was asking the question. Yeah, thanks
Right.
for that.
Okay, okay. Right. So we were talking about spectral libraries, right? So we said that if this is my material, so suppose this is granite from spectral library. This is band 1 and band 2. These are band 1, band 2 reflectance values. And then this is the uh, cell in my hyperspectral image. Like these are the band 1, band 2 values in my image. So depending on their proximity, I can tell if the cell has composition close to granite or any other end members which are available in the uh, spectral library. But in nature, uh, like not all the material compositions are uniform, right? So even if I'm looking at granite or concrete, it won't be uniform everywhere, it will have different values. So in this spectral space, instead of looking like a point, it will look like a cloud. So suppose this would be a cloud representing concrete, then there would be another cloud representing basalt. So instead of um, appearing as a point, natural materials, they would appear as a cloud in the spectral space. Okay, so in hyperspectral images, it is not necessary that, so these pixels have resolutions around like 30 meter by 30 meter, right? So it is not necessary that uh, each pixel would represent the material of the same type, like each pixel would represent the uniform material. So it might be like water bodies, it might be vegetation, it might be soil. Like it's not necessary that this entire pixel would represent a land cover uniformly, like water. So the spectral signatures that we receive from each pixel, they will be a combination of uh, spectral response of these different materials. So it, if um, this is for water, this is for vegetation, and this is for soil, then the resultant spectra would be like a sum of these three, depending on what fraction the mixing has been there in the cell. Like in this cell we have vegetation dominated area, right? So the spectrum would be the shape of the spectrum would be dominated by mostly vegetation, but then there would be inputs from water and soil also. So in order to interpret, to be able to interpret the image properly, we need to be able to deconvolve these different spectral signatures present in the like coming from the cell. So here we see that this curve C, it consists of 60% of response from A and 40% of response from B. So this is a composite spectrum, that is a linear mixture of two spectra, dry soil and green vegetation. And we talked about spectrum space in the previous slide, right? So we said that these end members would like they would appear in the spectrum space, so we can plot them as points. So if there is mixing between two or three end members, then in a two-dimensional space, if these are three end member spectra, these are the positions of three end member spectra, then after mixing, the resultant spectra would lie in this space. This is the mixing space. So n-dimensional plot of the three end member spectra for a hypothetical 2D band case, and all spectra that are mixtures of ABC alone must lie within this mixing space. Right. So this mixing can be either linear or non-linear. So in linear mixing, each reflected photon it interacts with only one type of surface material at a time, and then reflected energy it simply combines as an addition of responses from these different types of material within the cell. But then in non-linear mixing, what happens is a single photon it reflects with, it interacts with more than one material. So, like for example, if there is a uh, microscopic mixture of mineral particles in the soil, in that case the photon which is getting reflected it will interact with uh, multiple type of materials at the same time, so that will be an example of non-linear mixing. So this is an image from the lecture itself. These are the steps in hyperspectral data analysis. So first we saw that radiance needs to be converted to reflectance in order for us to be able to compare with the actual material on the ground. Then uh, there are these dimensionality reduction steps. So we'll look at these steps in detail in upcoming slides, right? Then we need to find end members using pixel purity index, and then we use spectral library to find the composition of these end members. So let's start with the first step. So this we had covered during multispectral imaging also. So spectral radiance measured by the remote sensor, it differs from the actual surface reflectance values due to spectrum of the input solar energy, interactions of the energy during its downward and upward passages through the atmosphere, the geometry of illumination for individual areas on the ground, characteristics of the sensor system. So these factors need to be accounted for while converting radiance to surface reflectance. Right. The next is dimensionality reduction. So why do we need to do dimensionality reduction in this case? So we had talked about dimensionality reduction during principal component analysis also. So in hyperspectral image, uh, we just saw that there are like hundreds of bands, right? So for each cell, so there will be uh, hundreds and thousands of cells in each image, and every cell will have corresponding hundreds of bands. So it would be very difficult to process this huge amount of data. And then, if the spectral response of the material is similar over a large wavelength range, in that case, most of the wavelength bands will contain similar information. So this redundant information it needs to be reduced, and there is some noise in the data which needs to be filtered. So for that we do this process of dimensionality reduction, so that it will be easier to do computations on this data. So imaging spectrometers in the adjoining table provide fine spectral resolution required to characterize spectral properties of spectral materials. So these are different types of imaging spectrometers, and look at the number of bands that they have. So here there are 224 bands, 286, 288, 211, and 128. So uh, these spectrometers are producing bands in hundreds of numbers, right? So this huge amount of data is generated for each scene. Yeah, as we discussed, adjacent wavelength bands might contain the similar information, and hence dimensionality reduction becomes essential to remove the redundancies in the data and reduce correlation between two bands while retaining the information content coherent in the imagery. So minimum noise fraction is applied for dimensionality reduction in this case. So this is like an slightly advanced form of principal component analysis. So this is used to transform. So this transform is used and retaining small number of noise-free components. So we'll look at the minimum noise fraction transformation. Right. So this transformation determines the true or inherent dimensionality of the hyperspectral data. So after uh, after removing all the redundancies and after removing the correlations between uh, two bands, there are MNF uh, transformed. So we have MNF transformed bands uh, which are generated, and then depending on how much variance these bands are covering, we can calculate the true or inherent dimensionality of this data. Like if out of some 200, around 20 bands are covering most of the variance, then that would be the uh, inherent dimensionality of this data. Then this transformation, it identifies and segregates noise in the data. It reduces the computational requirements of further hyperspectral processing by collapsing the useful information into a much smaller set of MNF images. Right. So this uses two sets of transformation. So first transformation, it decorrelates and rescales the noise in the data, and then second transformation, this is like in principal component analysis. So this creates a coherent MNF eigen images that contain useful information, and then it generates noise dominated MNF eigen images. Right. So uh, total information content, so these are the images uh, which are generated after MNF transformation. So total information content in these images after transformation, it can be checked either by visually inspecting these images or by plotting their eigenvalues or amount of variance uh, against their eigennumber, right? So these are the first 10 images after MNF transformation in this set of hyperspectral images. So this shows first 10 images. So after 9, 10, we see that the images are not differing as much, but then they're showing almost the similar information. And then here we see that check the cumulative percentage of variance. So here first 10 bands itself are covering about almost 98% of the total variance in the data. uh, Also here the eigenvalues are decreasing exponentially with uh, the eigen number. So first few images would cover the highest variance in the data, and then eventually it will be lesser and lesser. So this is MNF transformation applied on every run 8 scene. Right. So in this hyperspectral data analysis, first we saw that we converted radiance to reflectance. Right. Then we transformed our data by MNF transformation, and then removed the redundancies in the data, and removed correlation between the bands. Right. So MNF transformation. Now we have the total number of bands which contain the most unique information in this data set, right? So after this, the next step is to identify the end members. So we need to identify the most pure pixels in this image which can act as the end member. So uh, the mixed pixels will be linear combinations of these end members. So identification of these end members is an important step during this analysis. So identification of end member spectra to mathematically unmix each pixel spectrum to identify relative abundance of each end member materials. So this is why the identification of this end member spectra is important. Each image spectrum is modeled as a sum of fractions of the end member materials. So we just discussed this, and then best fitting set of fractions is found using spectral matching procedures. So we'll look at these spectral matching procedures in the next few slides. Right. So end member selection, it can be done directly from the image using field information or empirical selection techniques, or these end members can also be calculated using spectral libraries. So pixel purity index is one of the ways of identifying these end members. So n-dimensional scatter plots of the clean MNF images are projected repeatedly on a random unit vector. So and most extreme pixels in each projection are noted every time. So what happens is, we'll look at this figure on the right side. Uh, so after MNF transformation, we have different MNF transformed images. So this is uh, reflectance values in component 1, and then we are plotting them against component 2. Uh, so this is a two-dimensional space. So now uh, we have these many number of pixels in an image. So we have multiple pixels, right? So for each pixel, I will have a different number of uh, MNF components after MNF transformations. For each image, we have multiple MNF components. Now, for every pixel, So these dots are all the pixels in this image. So for every pixel, I am plotting component 1 against component 2. Or like we can extend this to a n dimensional space also. So this is a scatter plot. So these all these points, they are representing each pixel in the image. Now these extreme pixels, these pixels which lie on the extremes of this scatter plot, these pixels are noted down and they're marked in the image. So a random vector is taken, and on that vector projections of all these pixels is taken, and then this process is repeated multiple number of times. So um, uh, every time the extreme pixels are noted down. So each pixel, the uh, higher the number of times it is marked as an extreme pixel, the more would be the pixel purity index of that pixel. So this figure shows two component plot showing one of the random vector directions tested by the pixel purity index procedure, and all spectral points are projected to each test vectors and extreme points are noted. So now here this is the random vector. Then in some other case there can be, in a different iteration there can be a different random vector. So in this case, if we project all these points here, then again this will be the point at extreme on this random vector also. This point will be projected here, this point will be projected here. So this will also be uh, uh, extreme point on this random vector also. So these two pixels were extreme two times. So this is how we generate different random vectors and try to calculate how many times each pixel was categorized as an extreme pixel. Then that will be the pixel purity index of that pixel. And then we plot PPI maps of such pixels. So here, um, most extreme pixels in each projection are noted every time. Pixel purity index calculated for each pixel based on how many times it was classified as an extreme pixel. Right. Then a threshold value is determined to identify the purest pixels in the image. So uh, suppose this point was categorized as an extreme pixel 15 times. Then this point was categorized as an extreme pixel 7 times. So like this, every point will have a PPI value. Then we set a threshold that only above 10, only pixels which were categorized as extreme pixel at least 10 times, that is the threshold. So above that threshold, we will find all the spectrally pure pixels. So we'll look at this figure. This can only identify look. Okay. So pixel purity index PPI image of a portion of run 8 scene 5 derived from a masked minimum noise fraction Iris data. So pixels in A denote original output from the PPI analysis. So this first figure, it shows uh, all the extreme pixels which were found in this image, right? And then pixels highlighted in red, they represent a subset of those pixels selected using a threshold of 10 hits. So this image, all these red pixels, they are showing uh, the locations which were categorized as extreme pixels uh, at least 10 times, right? So these extreme pixels, they will be like they will be the end members in this scene. So they will be the most purest pixels. So if a scene has soil, water, vegetation, then these purest pixels will be uh, most likely to have all soil content or all vegetation content or all water content. So this is how we find the purest pixels or end members from an image. Okay. Okay. So there is n dimensional end member visualization. Uh, So this is a three-dimensional space. So n-dimensional visualization of four potential image-derived vegetation end members and one shade end member derived from PPI analysis. And then end members associated with other materials are not highlighted, and note that some of the highlighted pixels are not immediately recognizable as end members, but data cloud rotations were necessary to discern all the end member regions of interest. So here in a higher dimensional space, so before uh, in the previous slide we saw this same plot in a two-dimensional space. Now here, this is the same plot in a higher dimensional space. So in that case, the corners, the convex corners of these clouds, they would be categorized as the end member pixels. So corners of the data cloud are located in an n-dimensional space, and the purest pixels lie at the convex corners of the data cloud. The location of this data corners is compared with the location of the end members obtained using a spectral library either collected from the field or laboratory. And then this is how the pure pixels can be labeled as a type of end member. So uh, these end members, so before, if we do not know that each corner corresponds to which type of land cover, right? So we simply plot all the pixels in n dimensional space. We find corners of convex corners of this data cloud, and then those would be the extreme end members. So we know that these pixels, they correspond to some sort of uh, pure pixels. So they correspond to either like soil, water, or vegetation, or some other pure pixel. But we do not know that which corner is which pixel. So in order to find that, we have uh, like uh, an ideal or some reference spectra which is obtained either using field measurements or lab measurements or from spectral library. So these this image we have obtained using uh, hyperspectral data. So in that, from that data I have found locations of few end members in this n dimensional space. But then to find which corner corresponds to exactly which land cover, now we can do two things. One is we can use spectral library. So spectral library, so if I am looking for water, then this spectral library will have an ideal spectrum of water uh, already there. So if this is the spectrum, then we will take reflectance values corresponding to each band and then plot it in this n dimensional space. So suppose if water plot somewhere around here, then uh, we can interpret that this corner, so these pixels, they correspond to water. So this is first method, or using spectral libraries. Or second method is directly going to the field or doing experiments in the laboratory and finding out their spectral response. So we can use spectroradiometers. We can go to the field. We can check the land covers, different types of land cover. So in field we can check water, soil, or vegetation, right? And using spectroradiometers, we can generate a spectral response and then plot the same in our n-dimensional space of our remotely sensed images. And then accordingly, so suppose if this was classified as vegetation, then accordingly we can compare this field data with our remotely sensed data, and then we can assign or we can label these identified corners as different type of end members. So this is a numerical based on uh, spectral mixing. So you can try this for some time, and then I'll try to solve. All right. So we have end member spectral reflectance values in band 1 and band 2 for vegetation and soil. And then we have mixed pixel spectral reflectance values in band 1 and band 2. And it is given that vegetation and soil are the only two land covers present in this pixel. Right. So this is vegetation and this is soil. So suppose fraction of vegetation in this pixel is f1 and fraction of soil is f2. So in band 1, it is given that end member value of vegetation, end member reflectance value is 0.25 for vegetation, and for soil it's 0.7, right? And because vegetation and soil are the only two land cover types, we also have a condition that f1 plus f2 is equal to 1. So these two fractions, their sum is equal to 1. And it is given that in band 1 we have mixed pixel spectral, so the total spectral reflectance value coming from this pixel in band 1 is 0.55. And similarly for band 2 we have values zero point 0.4, 0.65 and 0.566. So we can solve this using one band value itself. So here we have f2 is equal to 1 minus f1, right? And here the mixed spectral reflectance value for band 1, uh, we have 0.55 is equal to fraction of vegetation is f1 into 0.25, plus we'll have f2 into zero point 0.7. And if we substitute f2 is equal to 1 minus f1, then we have 0.55 is equal to, so f1 is equal to 1 by 3 and f2 will be equal to 2 by 3. So even if we try to calculate for band 2, we should ideally get same f1 and f2 values. So these are the mixing fractions in this image. So vegetation is 1/3 and soil fraction is 2/3. So I hope the previous problem was clear. So let's try to look at different types of classification in hyperspectral images. So we first converted radiance to reflectance, then we did MNF transformation, then we calculated the end members or the pure spectra, and now we will see a different classification techniques in hyperspectral image analysis. So we have so there are two different types of classification: uh, hard classification or per pixel classification. Okay, so this is, there is soft or subpixel classification. Right. So in hard or per pixel classification, uh, each each pixel, it can only have one type of class. So uh, here this is each 30-meter by 30-meter pixel, right? So each pixel can only have one type of class. And in case of soft or subpixel classification, each pixel can be composed of fractions of different types of classes. So, like as shown here, each pixel. So here we see that these are different types of fractions of different classes present in this present in each pixel. So here we have 0.19 fraction of class 1, 2, 3, like class 1.19, class 2.25, class 3.56. But then here, so highest fraction is 0.56 of class 3. So here in hard classification, it is classified as class 3 itself. But then in soft classification, we can have different fractions. Yeah, and similarly, we can see for other pixels as well. We will look at uh, the classification type of spectral angle mapper. So this is a per pixel classification, right? So this algorithm, it takes an atmospherically corrected unlabeled pixel. So in an image, we have identified some end member pixels, right? So for those end members, which know we know that what these end members are corresponding to, but then other pixels which are not these pure pixels, we do not know that they correspond to which type of class. So it's unlabeled. So this algorithm, it takes an atmospherically corrected unlabeled pixel and then compares it with reference spectra in the same n dimensions. So this algorithm compares the angle alpha between the reference spectrum and the hyperspectral image pixel measurement vector t in n dimensions or n number of bands, and then assigns it to the reference spectrum class that yields the smallest angle. So we have these end member values, which are like our uh, reference spectrum with respect to which we will classify all the other pixels, right? So suppose we have uh, suppose there are different types of end members: uh, soil, vegetation, water, concrete, and so on. So this pixel, now it corresponds to it has each pixel, it has several corresponding bands. So we plot this pixel in an n dimensional space. So this is material T, suppose, right? And then there is a reference material R. So one of these materials, it's plotted here, and then there is another material which is plotted here. So in an n dimensional space, we have a vector joining origin to that of the point representing that spectra. So we will have two different vectors joining an unknown pixel and reference spectra of any of the land cover types. So we calculate the angle between the unknown pixel and the reference spectra, and then smaller the angle between the reference spectra and the unknown pixel, more will be the correlation between those two. So this is the formula: alpha is equal to this terminology, this expression. So here alpha is the angle between reference spectrum and the hyperspectral image pixel. So this T. So this would be this would be the angle between these two vectors. So how does classification work here? So there would be some threshold value of alpha, right? So if the angle alpha between the reference material and the unlabeled pixel, if it is less than that threshold value, then that pixel will be classified as that particular reference material. If it is more than the threshold value, then we will check uh, the alpha angle between this unlabeled pixel and other reference materials. So first, if suppose we checked this angle between material T, like the unlabeled pixel and concrete, so this value was greater than threshold, so it will not be classified as concrete. Then we will check same angle between all the other pixels. Then if for these pixels, uh, for these reference materials, if the angle is less than the threshold, then it will be classified as that particular pixel. So here uh, every pixel will have only one type of class associated with it, so it will be a like it will be a per pixel classification. Okay, then we have binary encoding, which is another type of classification method. So here, uh, so this is look at this figure on the right first. This plot. Uh, so this green curve, it is laboratory derived in situ vegetation, and the second pixel, sorry, second curve, it is remotely sense derived vegetation, and this third curve, it's water, it's remotely sense derived water. So why do we need binary encoding in the first place? Is that uh, same reason we have multiple number of pixels, and each pixel is corresponding to so many images, so many bands, and we need to compare each pixel with different spectra in the reference spectrum to classify that pixel, right? So it will be more easy if each spectrum, each pixel, the spectrum corresponding to each pixel, it is represented as a binary encoding, and then each pixel for reference material also we have the same type of binary encoding, and then we can just compare the results of this binary encoding to classify a particular pixel into as a reference material. So this is a reference curve of laboratory derived in situ vegetation, and this red curve and the water, these are remotely sensed derived values. So here we have a threshold T. So this threshold will be like an average spectral response of that pixel. So there will be a threshold value, and this is the algorithm for binary encoding. So if the reflectance value is less than the threshold, then the binary value will be 0. If the reflectance value is greater than the threshold, then this value will be 1. So let's look at different bands in this spectrum. So we have first 500 to 520 nanometer. So this uh, here, then 520 to 540, then 540 to 560, and so on, right? So in this first band, we see that water is less than the threshold. So water has value 0. Uh, Here, vegetation, in situ vegetation, like the reference material, and remotely sensed vegetation, both of them, they are also less than the threshold. So both of them are also classified as 0. Then in the next band also, they are less than the threshold. So they are classified as 0, 0. Now, in the third band, here, uh, lab in situ vegetation and remotely sense derived vegetation, both of them are greater than the threshold. So their binary value becomes 1. So here we see that both of them have value 1, and then water has value 0. Then again, uh, in after 700 nanometer, we see that uh, this lab derived in situ vegetation and remotely sensed vegetation, they have values greater than the threshold. So here both of them have values corresponding to 1, and then water is remaining below threshold in all the wavelength ranges. So water has value 0 everywhere. Now, this is a pictorial representation of these binary codes in situ vegetation, remotely sensed vegetation, and remotely sensed water. So uh, this is reference spectra. So here we see that uh, remotely sensed vegetation and in situ vegetation, their binary maps, they're showing higher degree of similarity than that of the remotely sensed water. So remotely sensed water does not show any similarity with that of the reference spectra of lab derived vegetation. But then remotely sensed vegetation is showing higher similarity. In that case, that pixel will be classified into vegetation. So this classification method is also per pixel method, right? So there is some index which is calculated to find how what is the extent of similarity between the reference spectra and the image spectra. So based on this, the classification is done using this binary encoding method. Okay, then next we have spectral feature fitting. This is also another classification technique. So this method is used to identify and map specific materials or substances based on their unique spectral signatures. Right. So this method, it aims to detect the presence and estimate the abundance of specific materials or compounds within a hyperspectral dataset. So before, what we were seeing is we were only looking at an unlabeled pixel, and then we were classifying it based on its similarity with the reference spectra. Now here we are uh, doing this classification based on the spectral properties of that particular pixel. So uh, if this is a particular pixel in that image, we are looking at the spectra of that particular pixel, and then we are comparing it with uh, spectra of the known reference material. And then using this image, we can also identify uh, abundance of different reference material in each pixel. So this method uses continuum removed pixel spectra and reference spectra. So we had discussed this continuum removal in the last session. So this is, sorry. So this is the overall shape of this spectrum. Then after continuum removal, uh, it will be like this. So after continuum removal, we can compare the uh, absorption bands in two different images. So this method uses continuum removed pixel spectra and reference spectra. Then depth and shape of the feature in the target spectrum is compared with that of the reference spectrum. So based on, so this is my unlabeled pixel, and suppose this is the target pixel. So based on the shape and depth of these shape of the spectral curve and depth of the absorption feature, based on this, this unlabeled pixel is compared with the reference spectra. And these scaled images are generated for each end member. Images show if each pixel contains a particular end member along with its abundance in the image. So if there are like four or five end members of, so we have soil, water, vegetation, concrete, right? So for each end member, a scaled image will be generated. So this will be, so if this was my original image, then for each end member, I'll have same image, like for every pixel. Here, every pixel will contain the abundance value of that particular end member in that pixel. So here, suppose most of this pixel is dominated by soil. So it will have values around, like if the values are ranging between 0 to 1, then the pixel dominated by soil will have values around zero point 0.9 or higher. Or so this is, the entire image is for soil only. So this is a scaled image. So then for every pixel, I'll see. Uh, so here, if soil abundance is less, then it will be slightly less. If here it's, here it might be zero, or so for every pixel I will have a soil abundance map, and then there will be similar maps for each end members. So this is spectral feature pitting. Then we have subpixel classification. So we saw that in subpixel classification we can find out fractions of each type of end member in uh, each type of end member in each cell. So this type of classification, it estimates how much target material is present in each pixel. Now there are different types of subpixel classification. First is match filtering. It maximizes the response of target spectrum within the pixel and suppresses the response of everything else and matches with the reference spectra. So uh, suppose in this image, My target spectrum is some 2 3 types of uh, end members. They are only my target spectrum, so I only want to calculate the abundance of soil, water, vegetation, and then everything else which is there, it is so the response of everything else will be suppressed, and then soil, water, vegetation response of these target pixels will be enhanced in this match filtering method. The second method is mixture tune matched filtering, or MTMF. So this method treats each end member independently, and at each pixel for each end member, it models the pixel as a mixture of end member and an undefined background material. So this MTMF outputs a matched filter score and an invisibility value for each end member. So this MF score is an estimate of the aerial coverage of a pixel by the material of interest and is analogous to fraction value from simple SMA. And the invisibility is a measure of how likely the pixel is to contain the material of interest. So lower the value of invisibility, more likely it is to contain the material of interest. So these are some of the examples from previous year assignments. So you can attempt these for some time and then we'll discuss. So these are based on hyperspectral remote sensing. So first is Dash is a two cascaded PCU. So this is MNF. So we just discussed that in MNF we use so there are two rotations uh, for during the dimensionality reduction. The role of Dash is to normalize the background information so that the individual absorption features can be compared from a baseline. So we just discussed this. This is continuum removal. This is a subpixel method used for maximizing the response of pixel spectrum while suppressing the background in the pixel. So this is matched filtering. The same material can show multiple spectral signatures due to change in composition. This is the next set of questions. So, this is the spectral classifier in which the scale of reference and pixel spectra must be the same. So, this is spectral feature fitting because here we after continuum removal we compare the depth of the absorption feature in image spectra and the reference spectra, right? So here the scale of reference should be same for both reference spectra and the pixel spectra in order for us in order to be able to compare. Like in contrast, when we looked at a spectral angle mapper, in that case, if this was reference spectra and this was pixel spectra, it would not matter if the um, scales of the two are different because we are only looking at the angle between the two of them, right? So here, even if the scales were different, like they could be it could be more or lesser than the reference spectra, the reflectance values. But then, because we are using the angle, the scale would not matter. But then, in case of spectral feature fitting, the scale has to be the same. This is the linear. This is the combination of mash filter and linear mixture method. So this would be the mixed tune mash filter, MTMF. A spectroradiometer's capability is described by. So this would be all of the above. So spectral range, signal to noise ratio, and spectral bandwidth. All of them are they all of them control the spectroradiometer's capability. Noise whitening in the MNF is performed in this rotation. So this is first rotation. We just saw this. Okay. Next we have support vector machine or SVM. So this is another classification method which is performed using machine learning. So SVMs find an optimal separating hyperplane between the classes using the training samples. So first, these training samples, this is okay. Uh, this is a set of data set which is already there and which is labeled. So these samples, they are plotted against each other. So this is a two-dimensional space against band 1 and band 2. Now we need to, in order for us to be able to classify an unknown pixel, we need to find a hyperplane which separates these two different training data sets. So this is training data set for class A. This is a training data set for class B. So in this case, we are looking for the optimal separating hyperplane between the two classes by maximizing the margin between the classes closest training samples. So here, this is class A, this is class B. Now there can be so many different planes here, right? So how do we know that which is the hyperplane? So this plane has to be such that the distance between these last points, so these are the uh, closest training samples to the hyperplane. So distance between them and the hyperplane is maximized. So better the classification, like better the position of the hyperplane, it will be easier to classify the unlabeled pixels. So the points lying on the boundaries are called support vectors, and the middle of the margin is the optimal separating hyperplane. So these are the support vectors, and this is the margin, which is the distance between these two support vectors, and this is the hyperplane which lies in the middle of this margin. So this figure shows a support vector machine classification, which relies heavily on the training class samples on the edge of the margin to identify the location of the separating hyperplane. Right. So we want the classifier with as big a margin as possible. Right. So higher the margin between these two classes, more easier it would be to identify or to classify different pixels into these classes. So the distinction between the two classes would be very clear if the margin is as large as possible. So distance from a point x0 y0 to a line is ax plus by plus c divided by this. So distance between h0, which is the hyperplane, and h1 will be given by this. So it will be inversely proportional to w. So w is the weight factor associated with that hyperplane. So total distance between h1 and h2 is 2 by norm of w. So in order to maximize the margin, we need to minimize w. So margin is 2 by w. So if we have to maximize the margin, w needs to be minimized. So with the condition that there are no data points between h1 and h2, we have following equations. This, which can be combined into this equation. So when it is not possible to find a linear separator, the data points can be projected into a higher dimensional space using kernel techniques. So let's take an example of 1D, of a one dimensional case, and then look at it in two dimensions. So So this is in one dimensional. These are two different classes. So this circles and squares. But in one dimension, we are not able to uh, find a plane which differentiates between these two classes. So we go to a higher dimension to find a hyperplane which distinctly separates these two classes. So if we project this into a higher dimension, we might be able to find a hyperplane which separates these two classes. So these SVMs focus on solving the optimization problem with training samples using following equations. Uh, so these are the equations, and this is the positive slack variables used to allow some of the samples to fall on wrong side of the hyperplane, and this term is used to penalize solutions for which this is very large, and this term is a hyperplane in a highly dimensional feature space. So these are few of the previous year assignments based on this part. So linear classification of datasets in the feature space is optimized by SVMs. The quality of the spectral library does not affect the accuracy of identification and abundance estimation. So this will be false. What properties of the spectrometer are most essential for better spectral library creation? So this will be high spectral range and narrow bandwidth. So um, higher the uh, spectral range and smaller the bandwidth, will be able to characterize the material in a better way. The reflectance spectra are influenced by the source sensor geometry. So this is true. Spectral feature fitting is based on absorption feature mapping and base suited hyperplane for separating the given classes in support vector machine method is. So here four different hyperplanes are given. So y would be the best suited hyperplane. Which of the following shows the comparison of the pixel with the reference spectra by observing the angle between them? This is spectral angle map. And which of the following is a data dimensionality reduction technique? So we have seen PCA and MNF, both of which are data dimensionality reduction techniques. ICAS, independent component analysis, which is also a dimensionality reduction technique. So this would be all of the above. Now, after this, there was applications of the hyperspectral remote sensing data, which was pretty clear in the lectures itself. So I'm not going to repeat that. So we'll start with the geographical information system. So a geographical information system, it consists of integrated computer hardware and software that store, manage, analyze, edit, output, and visualize the geographic data. So this is how a GIS system enables us to look at various layers in a georeferenced image. So we can look at various layers together. So like flood zones, parcel, land cover, elevation, and then all of these layers, different layers, can be plotted together in a GIS system. So, GIS mapping produces visualizations of geospatial information, and then the four main ideas of a GIS system are to create a geographic data, manage it in a database, analyze and find patterns, and then display it on a map. So, because viewing and analyzing data on maps impacts our understanding of the data, we can make better decisions using GIS. So, GIS enables us to like spatially visualize the data. So it really helps us like understand the data better when it is plotted on a map. uh, yeah, it helps us understand what is where, and the analysis becomes simpler and the answers become clearer. So these are the different components of a geographical information system. So first is data, there is data, hardware, and software. So GIS stores location data as thematic layers, and each data set has an attribute table that stores information about the feature. Right. So this data, like as we saw in this previous figure, so this would be the data about flood zone. So it would be either a vector or a raster layer. So if this is a vector layer, then it would be consisting of different polygons, right? Or it could be a raster layer consisting of a small pixel, and then each pixel will have a corresponding spatial information, and uh, it will have a dn value associated with it. So we can visualize these layers in a GIS system. So we can visualize all such different layers together in a GIS system. So it stores location data as thematic layers. And then there are two main types of GIS data, like a raster and a vector. So here a figure, we look at figures on the right. So this is raster data. So it looks like grids because they store data in rows and columns like this. So here each pixel will have a corresponding lat, lon, and then a z value. So it could be an elevation value, or it could be a brightness value, it could be surface reflectance value, depending on what data we are looking at. So these data can be discrete or continuous. Like for example, we often represent land cover or temperature or imagery as a raster data. So next is second type of data. So raster data can have formats like .tiff. Then we have vector data sets. So vectors are points, lines, and polygons with vertices. So this is a polygon data. There can be a point shape file. So like we can look at locations of, for example, all dams in a region, or all groundwater monitoring wells in a region. So these points will be, this is a vector file. So each point will have a coordinate information, and then it will be saved as a shape file. Or there can be lines. So uh, rivers or roads uh, can be at in a very large scale map. uh, these can be shown as line features in the image. Like it can be saved as a line layer, vector layer. uh, for example, fire hydrants contours and administrative boundaries are often vectors. Or there can be polygon files of lakes or uh, gardens, forest areas, anything. Second component is hardware. Hardware runs GIS software. It could be anything from powerful servers, mobile phones, or personal GIS workstation. It could be anything. And the third component is software. GIS software specializes in spatial analysis by using math in maps. So it blends geography with modern technology to measure, quantify, and understand the surrounding world. So different examples of GIS softwares are QGIS and ArcGIS. So QGIS, ArcGIS is licensed, but QGIS is an open source software. So we'll stop here for today. Maybe in next class I can show you a demonstration of QGIS and different other components of a GIS system. So uh, yeah, that's it for today. So please let me know if you have any questions, and this time we'll meet on Tuesday itself. Are there any questions? Okay, thank you everyone for joining.